Hey Education fans, this tutorial is going to cover transcription and RNA processing. So here we are with our happy-go-lucky double-stranded DNA. Again, a snapshot of our chromosome. We will denote a section of it to be a gene, a coding region that will create mRNA and then protein in the very end. So what we have directly upstream of a gene is called a promoter region. And in eukaryotes, this region usually has a Tata box at about minus 27 from the +1 of transcription. The +1 of transcription, right, will occur at the very beginning of the gene. So what happens, without drawing a whole bunch of proteins in here and making it a big mess like we did in class, is the Tata binding protein, right, binds to the Tata box. So a protein physically grabs the DNA around that sequence. That brings in RNA polymerase, and RNA polymerase, another protein that grabs TBP and the DNA, and then begins complementary base pairing off of the template DNA. Now remember, the non-template DNA is the one that has an ATG in the 5' to 3' direction, which would mean there's a TAC on the actual template strand. So if you look for the ATG in the non-template, it's the other one that's a template. And so we then would begin transcribing, doing complementary base pairing, adding ribonucleotides. And so I'm going to draw that as a single line, because it's single-stranded. Now, remember, the polarity of mRNA is always denoted the 5' end on the left and the 3' end on the right. We always list nucleic acids from 5' to 3'. Okay, so this is what our book would call pre-mRNA. Not mRNA, pre-mRNA. I like to refer to it as a primary transcript, but I think our book uses pre-mRNA. So this pre-mRNA is not what's translated. Okay. First, we have to process it. So the process of going from, backing up a little bit, the process of going from DNA to RNA is called transcription. So there's transcription, right? Complementary base pairing on our template DNA to create our pre-mRNA. The next thing we need to do is called RNA processing. In RNA processing, I need a color. We need to do 3 different things to our RNA. We need to put on a 5' cap. So here we go. Let's go back to our RNA strand. Okay, we need to put on a 5' cap, which I'm going to put in orange. And the 5' cap is just what they call it because you think about it like a hat on the 5' end. It's a guanine ribonucleotide that's methylated. Okay, so I'm going to show that guy as a G with a methyl group attached to it. Okay, that is bonded upside down to the first nucleotide in our pre-mRNA. And so I'm just going to write a couple of those in there just to make it a little easier to see. So we'll pretend like our first base is AAAA, CCC, and so on and so forth. Okay. The next thing we need to do is add a poly-A tail. And we'll call that the cap. So what we do to put on the poly-A tail is essentially phosphodiester bond a whole bunch of adenine ribonucleotides to the end, the 3' end of our RNA. These can be hundreds of adenines. These are known as the poly-A tail. So we have to process, we had the cap, we had a poly-A tail, that guy. Okay. And then there's one more thing we need to do before we can call this an mRNA. right? So this is still pre-mRNA. What we need to do is remove the introns. So before I show you that, we got to talk a little bit about how a gene is made up. In eukaryotes, genes are made up of segments that we consider coding and non-coding. Any region that codes that actually has some nucleotides that are going to end up in our amino acid in our real RNA that will then code for the amino acid sequences are called exons. Anything that doesn't is called introns. So introns and exons make up the genes. Okay, so the entire thing is transcribed, right, all the way across from beginning to end in our pre-mRNA. We add a cap and a tail, and then we need to remove the introns. So we can sort of follow our thought process here. Okay, and so this would be an exon, this would be an intron, this would be an exon, this would be an intron. Again, down here, right, exon, intron. And when we go to splice, right, cut these out, physically remove chunks of ribonucleic acid, we will end up with this guy is on here. Right, we have the 5' end and the poly A tail. Okay, and this is an exon, this is an exon, and what essentially gets spit off are two little pieces of RNA that were the two introns that were in that gene. And now we can call this guy mRNA, and we can send him out to the cytoplasm to be translated using the sequences in just the exons to help us choose what amino acids in which sequence should be into the protein. So this is for eukaryotes, right? So let's make sure we realize this is euks up here. So this is eukaryotic transcription and RNA processing. If we go to prokaryotes, right, I'm going to try to, right, we have one circle and it's double-stranded, right, there's a whole bunch of genes on here. I'm going to say here's a gene, okay, and I'm going to tell you this region's the promoter. What do we have to have in there? Remember the -10 and the -35. Then what happens? Sigma factor binds. Then what happens? RNA polymerase comes in. Right. Then what happens? We get the start of transcription at the +1. And there we go. We get transcription. Here's the 5' end. Here's the 3' end. This is transcription. And guess what? We are done. In procs, that's all they do. Immediately, this mRNA can get translated. Right? Doesn't have to go anywhere because prokaryotes don't have a nucleus, so they're in the cytoplasm already, and ribosomes can just hop right on. So there is no RNA processing in prokaryotes, right? So if we go back here, we can look at that. So if we write RNA process and put this, big cross through it. So the original RNA that comes off from transcription is the mRNA in prokaryotes. In eukaryotes, it's not. In eukaryotes, we get a pre-mRNA, we have to cap it, splice it, stick a poly A tail on it, then it's mRNA, and we can send it to the cytoplasm. That's your tutorial for today, and I will talk to you soon. Thanks for listening.